symposium on the future of snake research and conservation. I'm sitting here today with four really interesting people, and as you might imagine, this recording is going to play on the Snake Talk podcast, so you'll be able to get this on Spotify, Apple Podcasts, YouTube, any of the places you digest your podcasts. And the way we're going to do it is I'm going to introduce each of these people shortly, but we're going to have kind of a topic-based discussion where I interact with each of them around an aspect of, again, the future in snake ecology and conservation. And we'll kind of go back and forth and have a little bit of a discussion, maybe somewhere in the ballpark of about 10 minutes. And then we'll do that with each of the guests. And then we'll open up to the audience, and we'll have somewhere in the ballpark of maybe half an hour session where the audience can ask the panel questions. And if you don't have any questions, I'm sure I can talk for a long time. So I'll come up with some more questions for you guys. So first of all, I would like to start off by introducing all of these fine people here to my left. And I've never met Lisa until today, but Lisa Hallock, she's a herpetologist with the Washington Department of Fish and Wildlife. And she's going to be talking to us today about natural history. Then we have Dede Olson, who most of you probably heard speak recently. We're going to go a little more in depth on snake fungal disease. I've known Dede now for, years. I don't know, a long time. I started my PhD up in Vancouver, British Columbia at UBC, and there was a little bit of a financial crisis trying to survive in Canada. And so I came back down to the States, and Dede was nice enough to hire me. And so I actually worked in Oregon probably 20, maybe 25 years ago on a salamander cruise, working with Dede, before I ended up moving on to Idaho State and doing my PhD with Dr. Peterson. And did I already say we're going to talk about snake fungal disease? I did say that. Yes. And then Lameace Hussein, they are a PhD student at the University of Florida. And this is not her first time on the Snake Talk podcast. There are other people in the room who've been on the podcast as well. But you can hear Lameace's episode is episode 68, where she talks about human dimensions, DEI issues in this field that we're in, so I'd encourage you all to listen to that. And we're going to talk a little bit today about outreach. And then in the end, we have Dr. Steve Mackessy, who's a professor in the School of Biological Sciences at the University of Northern Colorado. Many of you probably know him, and he is an expert in Venomics, and we are going to get a little more in-depth on that today as well. And I will say that this is all being recorded, and again, will be on the Snake Talk podcast. So when we do get to the discussion and question and answer session, we will actually, Tracy here, who works with the Oriane Society, she will be moving a microphone around, and you'll answer the question into the microphone, and that will record it and get it onto the podcast. And also Mark's running the right side of the room, in case the one person over there has a question. So, okay, great. Do you guys have any questions for me before we start, panelists? Okay, well, let's start with you, Lisa, and let's talk about natural history. And, I think natural history, no matter what discipline of science that we are in, all of us probably have some level of appreciation for natural history. But I think it also, over time, maybe has a reputation with some people of almost being like a little bit of like a soft science. And so maybe we can kind of kick off this discussion by your thoughts on maybe how we could like resurrect this value to natural history in everything we do.
All right. Well, I think we need to start by having an agreement that we need that kind of information, and therefore, if we need it, that it's valuable. And so then, if it's valuable, what do we do both to get better funding for it and to have better ways to share that information. So one of the problems is it can be hard to publish that information, and so if you've collected it, how then do you get that information out to your colleagues, to others, so that it's usable?
And a lot of our jobs, that's one of the metrics, is how much we publish. So you talk about that basically maybe the reward for doing natural history research, and reward being some of the funding that comes to it, is matched with its importance. So how, in your agency, first of all, I guess I'd ask you, do you see that appreciation within your agency, and do you see a move to value natural history, to fund natural history in your state, or is that something you think really needs to improve?
Well, I guess I'll say, and this is a snake conference, so that sometimes the natural history information is the only information we have. Particularly with rare snakes, you might not have that many encounters with them. It might be hard to find lots of them in order to do experimental studies or more in-depth studies. And so also inventory, where are they? How many are there? That is important information to us to determine status, where we need to put attention, where we need to request information from colleagues, from the public. And so it is the starting point of kind of how we get a sense of what's happening with these animals.
Yeah. So do you have a sense if, so you obviously work, you probably cross a lot of boundaries with your title, meaning you're a herpetologist for the state of Washington. So you're probably working a lot with other scientists, with other taxonomic groups. You're working with people on the ground managing state properties, probably managing federal properties. How do you see those people putting value on natural history and incorporating it into the work they're doing? Or is that something, again, I'm kind of hammering on the same point, but I'm just kind of dancing around it to try to build a picture here.
Yeah, I guess it kind of depends. Certainly at this conference, we've had a real mix of experimental and observational, and so in this format, there was lots of that information shared. And so again, it kind of depends on funding source, also, again, how little we know. Sometimes we're starting from the very beginning, trying to figure out what's going on with these species. Also with recovery, we need to know the most basic things, phenology, what kind of habitat they need, where they're breeding in order to be successful in some of that work. And then, of course, as that grows, getting even more in-depth work and design to experiments.
Yeah. Do you have maybe a good example for the audience, maybe from Washington here where you work, where you think we've been able to take natural history, give it the warranted value, and you just see the natural history information kind of put on the ground, say, to manage or conserve species? Do you have an example from Washington that you could kind of give us an example of?
Well, I presented information on the striped whip snake, and that information that we've been able to collect is pretty basic. Inventory around the state, and then answering basic questions about the snakes. We've used that information to try to establish the areas that need to be protected and some of the habitat that we need to protect and some of the restoration work that needs to be done to make that habitat better for the snakes as we have invasive weeds come in and things like that. So that would be an example.
Great. So you think the whip snake's a good example. Okay. Let's move on to Dede for a little while. And Dede, I really want to build on the presentation you gave not too long ago. But I'll start off by saying that at the Orianne Society, so first of all, we're based in the southeast, based kind of right on the Georgia-North Carolina line. And we do a lot of work on snake fungal disease. We've been surveying prevalence for those species. We've sampled thousands and thousands of snakes across the Southeast. And what really struck me about your presentation was kind of this wave of, seeing all of this prevalence of snake fungal disease in the East, just like we're observing in the Southeast, and kind of seeing it as a wave, and it's maybe on the horizon for, for the Pacific Northwest region. And I want to get to that, and what I'm really getting towards is being proactive, and I want to hear more about this kind of strategy that you talked about. But the first thing, I want to put you on the spot, and I want you to speculate. And, so something you mentioned in your presentation, what is your gut telling you? So there are people in Georgia who are sampling thousands of snakes a year for this. And then, like you said, in Washington, this is just something, there's almost no surveillance right now. So do you really think this is something like with white nose syndrome that we saw truly moving across the country in a wave? Or do you think this is like a detectability issue that we're just seeing it where we look for it?
So, Chris, that's a really perfect question, and there's no answer for that because we don't know the distribution of snake fungal disease right now. But that's true. There is very little surveillance in much of the Northwest here. We're in Washington State, Oregon. I'm not aware of very much surveillance. We don't have detections in these states. Is it because people haven't looked and sent their samples in or analyzed it? Or is it truly a wave and it's not here yet? So an awareness needs to grow. So we're not looking because people may not be thinking of it, or they're not seeing these lesions on the skin of these animals, or they're not looking for lesions, and they might be there, but they just are not aware. So raising that awareness is needed. But we're in this opportunity to set up a strategic plan to do not just surveillance, which is needed since it's hardly happening, but to also look to see, well, where are we with snake information, snake health and snake ecology, just general information for our snake populations. And we're quite nascent in some respects in that regard. We don't know exactly where they are, or very few population level studies. So the ecology is not fully fleshed out at this point. For snake fungal disease, there's quite a bit going on with epidemiology research and diagnostics right now. We don't fully know the suite of fungi that might be contributing to a broad set of snake fungal diseases, plural. It may not be a singular disease. And so there's different aspects that we can think of in a strategic way, and I'm reflecting upon the situation of amphibian diseases, where BD hit, we didn't know about it until very much after the fact. There were enigmatic amphibian population declines being detected, and later one of the reasons for some of those turned out to be a fungal disease. That has informed both the white nose syndrome strategic planning, as you alluded to with bats, but also the strategic planning for B-Sal, a second chytrid fungus that is known from Asia and apparently has invaded Europe, and it woke up North America. And we need to develop a strategic plan to what are we going to do if B-Sal were to come to North America and affect amphibians. So thinking of what the B-Sal, the North American B-Sal Task Force has stood up, is a good model for snake fungal disease, I think, where they have different teams of different people with expertise looking at surveillance, research relative to the ecology. What does it mean for an individual if they're infected, or a population if they're infected, if anything? Some species can get these microbiota on their skin and they're not, that doesn't lead to a disease, for example. There's epidemiology research. There are quite a bit is needed for fungal diseases to make the diagnostic laboratory techniques more effective. There's uncertainty in the diagnostics, and that needs to be worked out. But it goes beyond that. The managers need to be made aware, and there's a management and control issue. What do we need to be thinking about ahead of time to control an outbreak if that were to be needed? How can we stop transmission if there's an element of human-mediated transmission of these pathogens? Are we transmitting it by handling animals, walking between sites, that sort of thing? So there's a management and control issue. And as we work with managers, oftentimes they like to see sort of these if-then scenarios, and that gets to decision support systems. If they don't have it, do they do biosecurity? If it is found, what do you do? Do you quarantine a site? Do you treat a site? What do you do with the animals at the site? So having those scenarios planned out ahead of time is strategic planning, and the managers really appreciate that, and they can provide feedback for that. There's recent evidence for snake fungal disease that supports that it could be an introduced species. And so that's a paper by Ladner et al., 2022. And so they have found different clades or strains of snake fungal disease of the Ophidiomycosis. And they have done the genetic work to support that it has been introduced multiple times to the United States. So that doesn't mean that it is an introduced species, but there is some scientific report pointing to it being introduced, and that would support this wave perhaps going from the east to the west of North America, and explain why it has been recognized as a big problem in causing dead animals, if not die-offs in populations in some selected species in the east, and we haven't seen that in the west. If it were to be introduced, then can we stop it? But I think the most important thing for our region in the Northwest, Washington and Oregon, and similar jurisdictions around it, is to start the communication and outreach now. This is a thing, and we need to be aware of it in the wildlife community. The managers need to be aware of it, and I think it's time to get ready. Strategically, we can work on all of these different elements, and it's not just a science community, it's not just the management community, it is enthusiasts for nature, and it can involve everybody. And everybody has a stake in this. It really is an issue, it's a new issue, and it can integrate different sectors of the society, and I'd like to see it go that direction.
So first of all, you're obviously have been doing public speaking for a while. You're a scientist because you avoided my my efforts to get you to speculate. And
You can try again. He says I didn't answer the question.
No gut feeling. Come on. No, I'm just kidding. I know it's impossible to know at this point. So with this concept that the snake fungal disease may have been introductions and potential multiple introductions, do we have any insight into where it may have come from, geographically, certain industries?
Well, in that paper by Ladner et al., they had a confidence interval of when it could have been first introduced to North America. And I don't know the exact dates off the top of my head, but it was something from the early 1830s through 2010 introductions through that range of time. And there is one clade, one strain that they focused on that had appears to have a more recent introduction from 1985 to 2010 or so. And so you can think of, what would be the mechanism of introduction in those, certainly people were moving quite a bit in those time frames. The first known incidents of, sort of a live snake being detected with Ophidiomycosis was on the east coast of the United States. And then if you look at specimens that were preserved and look to see when they were collected and infected, and you could tell that now that we know that, that infection was ophidiomycosis, when you look at the genetic diagnostics of those samples, that was also the eastern United States. So there's it's like we have some crumbs coming and it's supporting that eastern introduction that would support this wave going west, but we still have a lot of uncertainty.
Okay, so I like this concept. I like where you are, say, versus where we are in the Southeast, and that it's very prevalent in a lot of species. There's some bright spots from what we're learning there. Some species seem to do relatively well with it, and other bright spots. But I like where you sit in that you really do have an opportunity to be proactive. And so you mentioned this concept of being strategic, and you just mentioned some of the potential components of that and presented some of those this morning. So how do we get from that, from you saying we need to be proactive about snake fungal disease, and then you've just got so many entities and municipalities and researchers and agencies and universities. How do we turn this into a global strategy that that can be implemented?
Yeah, that's a huge endeavor. But I know that there are people that are actively working on this question, that they are engaged already, and some of those fit into the slots of being a researcher or a scientist or doing the surveillance as you're speaking. But I did attend the Global Amphibian and Reptile Conference in Knoxville, Tennessee. That was back in August last year, 2022, and there were people from around the world, and a dozen to 20 different diseases were presented, but the snake fungal disease caught everybody. And I think that there are certainly people interested in this topic from broad geographies. And so I think that it is possible to get a network of people going at that broad scale. But I think more importantly for North America, to get a group of people that are connected and interested in working on these different sectors is needed here, because it's certainly something that we're starting to track and follow. In the earlier session today, in the Snake Symposium, in the Q&A after my talk, I know there was a lot of passion for getting some biosecurity out there. So some proactive things, flyers at places that people go, the public goes, to make them aware of the issue. Perhaps biosecurity protocols for researchers, make them aware that we're going towards an era where you may need to be wearing gloves beforehand between each animal that you're handling, or disinfect between sites as you're moving across the landscape, or even for the public as they're going into an area that is managed for biodiversity, such as national parks, that they may need to step into a bin of bleach water to, even as a symbol, symbolically showing that there's an attempt to disinfect and not bring pathogens in from somewhere else that they have been.
And so, just so you know, we already do that in the Southeast.
You do that in the South, so let's get that going, especially for our most sensitive areas that we're trying to maintain the natural heritage of our biodiversity. If those areas, like our natural parks, they're open and we want people to go there, but we want to keep them safe for the organisms as well. So it gets to a snake health thing, and I think we can stand up some of those measures very quickly because they've already been conceived and developed for other emerging infectious diseases, such as the ones I mentioned earlier for amphibians. So very quickly we can have the disinfection protocol ready and get out the procedures for what the casual observer might be able to do versus the scientific researcher that may be handling a lot of animals, and perhaps transmission of snake fungal disease within a population is at issue, and gloves might be needed. So I think that there's several elements of this, but it's going to take a community to get it going, and we need to initiate that community.
Great. Lameace. So let's talk about outreach. And I think outreach is an interesting topic because, Lameace, can you pull your mic a little closer to you? There you go. So it's an interesting topic because I bet almost anybody at this wildlife conference, if you were to talk to them, they do outreach in some form, whether it's related to snakes or turtles or wildlife. But it's also really such a broad category and can take on so many different forms. So how do we define this thing? And one thing that I've found interesting in meeting you is that I think you're working, you're pushing some of the edges of it. And so I think it'd be interesting to kind of hear some of your thoughts. And so I guess I'd like to start out talking about where, in this big, broad mess, this word outreach, so where do you see, again, we're talking the future of conservation and research, where do you see, the real opportunities that maybe we haven't or are not currently taking advantage of in this big outreach space? And then also kind of a second part of this question, if we think about people, individuals who have been taught, or certain communities of people that just don't interact with snakes, or they've been taught certain things about snakes, how do you deal with that issue too? So it's kind of a two-part question. What do you see as the real opportunities, but then focusing in on people that may have almost a negative perception of snakes, how do you think we deal with some of those issues?
Yeah, so as far as some of the opportunities, I think a major one for me, I'm a really big advocate that every scientist should be able to communicate their science to the public. And I really despise the term dumbing down your science for the general public, because that's not what you're doing. You're essentially expanding your vocabulary so that people can understand it. And I think really one of the bigger opportunities is taking conferences like this and taking talks and being able to shift those in a manner that the public can see. And so as I've sat through all of your talks, I've been thinking to myself, like, okay, if I was creating an illustrative book for a land manager, for elementary school kids and stuff, like, how would I interpret what you're doing so that they can get just as excited about, your science and all your findings as everybody in this room gets? And so I think really bridging that gap and understanding that we are in a very small bubble at conferences. And so how we kind of expand that out so that the public can understand the science that's, going on. I also think that every person should experience handing a child a snake for the first time and seeing them scream initially and then light up as they, are able to kind of like face this fear of snakes. And to go into the second part of the question, in Florida, for instance, in the South, there is definitely a very bad perception of snakes. We have things like Burmese pythons down there that people are afraid of. Eastern diamondbacks are a huge thing. And the thing that I have found that works is we are lucky enough to have indigos in certain parts. And we have essentially, in a lot of rural communities around Gainesville, we have dubbed indigo snakes superhero snakes, right?
They're good snakes.
Yeah, they're the good snakes, right? And like that's the idea, is like when we get people who they're afraid of rattlesnakes, and when you talk to them and really understand like where this fear is coming from, you understand that a lot of it, like especially in lower income communities, comes from like fear of they get bit by this thing, they don't have medical access and stuff, they've got kids running around. And so we're like, oh, well, if you need a helper to get rid of your rattlesnakes, you should think about making, the areas around your neighborhoods and your house suitable for indigos. And we bring indigo snakes out as outreach animals. They're these, I'm sure everybody in here knows what an indigo snake looks like, but very big, beautiful snakes. And seeing even parents, as they watch their kids, handle these snakes and stuff, seeing, those gears kind of turning of, okay, well, if I can at least, have this snake on my property or this snake close to me, and I know that, my kid is safe with these snakes, then maybe, not all snakes are bad. And I think we focus so hard on not wanting people to kill rattlesnakes, and it's a super important thing, but I think first we have to shift focus to getting people to understand that not all snakes are dangerous first, and then you gradually work up to not killing rattlesnakes. But people are complicated.
Yeah, we do the same thing. We work in similar systems with indigo snakes and diamondbacks, and we certainly use the indigo snake as almost like an ambassador. And I talk to people who kill a lot of rattlesnakes, even though I've worked on rattlesnakes for so long, and I don't persecute them for that. I might talk to them a little bit, but how about so in terms of again, this outreach is such a big topic, and there's so many components, and there's so many ways that we could potentially spend our time as professionals working in the space. Do you have any thoughts on certain ways that we could maybe have the greatest impact for our investment, whether that's, who we might work with, how we might work with them? You mentioned kids, for example, kids versus adults, inner city versus rural kids who grew up in the how can we, and I'm sure a lot of you had this, but just personally, I get asked to do hundreds of things a year, and so I turn down so many now, and I just pick a few. I'll go do the scout group, and I'll, how do we prioritize to have a impact?
That's really dependent on what your goal is. So I always encourage people to get to know as many science communicators as you can. And if you are not comfortable going into maybe a super urban area in order to talk about snakes because you're I don't know if these kids are going to be interested in what I'm saying, there's plenty of community groups who, if you provide them the means and the information, will go out and do it. Also, especially kids now and their parents, social media. I know some people really hate social media, but social media has been a really big platform, especially when we talk about minority groups getting interested in herpetology and in sciences. And we see a lot of engagement on a lot of those platforms. And so I'm not asking any of you all to do a TikTok dance for snakes or anything, but hiring somebody to really put out these graphics, and essentially, like I said, taking the science that you have, that you presented here, and putting it on a platform that makes it really digestible to the public. And that, that's something that can be shared around, so you don't necessarily have to go in person. We also know that with high school students, elementary school kids, you can tell them anything and bring in a fake snake and they think it's real, so they're easy. High school students is where we really see the big barriers happening. And working with local zoos and even reptile zoos and stuff like that really helps for that, because those kids need an animal. Some of the best outreach that we've done with high school kids is bringing in Burmese pythons or these 12-foot snakes and just pulling them out of a pillowcase and just watching these kids just lose their mind for the first time of seeing an animal like that. And again, they're afraid of it at first, and then they get over that fear. But we know that programs like that really change trajectories, especially for high school students. And so it's working with science communicators. If you're not good at communicating your science, somebody's good at it for you. It's also working with teachers and school programs and stuff like that, and a lot of after-school programs to really bolster that type of outreach.
Yeah. And I do think it's important. So, I mean, I love putting a snake in a kid or an adult's hand. It's really powerful. I'm sure most of you have probably done it. But I'll tell you honestly, as I'm not necessarily the most technologically advanced person, but as time changes and I realize I want to keep up with that, you know, we've been revising how we do outreach. That's why we even have this podcast. I very quickly realized, each of these episodes reaches thousands and thousands of people versus I can go to a scout group and reach 15 kids. I still like to do that, and I still do quite a bit of it, but we're really trying to think innovatively about how we use technology and how we use it well to reach a lot of people, because in most cases, you know, through a podcast, for example, we can't put that python into their hand. So, okay. Well, let's leave it there for now, and we'll come back to everybody in the questions. So, Steve, in your presentation, you talked about how Venomics is that how you would pronounce it? Venomics?
Yeah, that's correct.
So how Venomics kind of influence or just gives us insight into natural history of snakes, rattlesnakes in particular. And so just to kind of turn that around, so obviously there's a lot of natural history information that we don't know for a lot of these species. Snakes in general are very hard animals to study. So do you see major gaps in, kind of our understanding of natural history? And if we filled some of those, how might that kind of improve our understandings of things like venom composition and things of that nature?
Sure. Well, let me back up a few steps here and put things into context for you. As an undergraduate, chemistry was the bane of my existence and certainly was hard on my GPA. It wasn't until I started graduate school that I realized that venoms are chemicals and chemicals involve chemistry, and so got into that end of things. So I started out with a very intense interest at the organismal level, and I've maintained that all the way through. And so when we first started working with venoms, we're looking at the venoms from trying to take the perspective of the snake. That is, we've got something that may consist of 100 different types of proteins, of 15 to 20 different protein families doing all these different things, and why are they so complex, and what is it doing for the snake? And so as things have progressed since that time, I was an undergrad during the late Pleistocene, I think, and so since that time, tools like mass spectrometry have kind of paralleled in development with the genomics revolution as well, and that allows us to take small amounts of material and really probe it very, very deeply for what is in that venom. And so now we have the capability of taking apart these venoms and finding out, vanishingly small quantities of material that are in here. But what is interesting to me still is to look at broader patterns. And that's where we're taking a look at the Venomics end of things, where we're trying to describe all of the different proteins in a venom. But if we look at broad patterns, and I talked a little bit about this in my talk, is that we see reiteration of these patterns. And these reiterations suggest that there's some commonalities that are being done amongst certain groups of animals that are using a certain type of feeding strategy, for example. But then we see some very much outlier groups that have a venom that's quite different. And then you've got some that can't quite make up their minds. So the Mojave rattlesnake has populations that are, some are type 1, some are type 2, some are in between those right in there. So it's fascinating from a lot of different perspectives. And what that also leads to is that it can, in fact, influence things that people care a little bit more about, and that is what to do if I get bitten by a venomous snake. And so as we understand more and more what the patterns of composition are, what we can expect to find in this population or that population, we can get a better handle on perhaps treating the nuances that arise for a particular snake bite. And people tend to be more interested in things that are within the realm of their experience. So along that end of things, though, we've found that when we're working with snakes in the field, that if we can bring in examples that are in the context of the realm of experience of people in a positive sort of way, that even things like snakes, and in particular venomous snakes, which aren't warm and fuzzy and cuddly and they're not charismatic megafauna of any sort, actually can change their minds. And one example of that is there's a compound in viper venoms that we mentioned briefly, disintegrants. The snakes seem to use that as a molecular tag for saying, this is my mouse that I envenomated, this is how I relocated. Well, it turns out that same compound has potentially and actually been shown to be anti-cancer in many respects, and that is that it's not a cell-killing agent, but what it does is it minimizes cancer cells' abilities to become metastatic. And that's where cancers become most dangerous, right? And so if we've got something we can keep localized, then perhaps we can use lower levels of chemotherapies and radiation therapies to kill off the tumors and things like that. We actually saw this demonstrated in the field. So we have worked with prairie rattlesnakes for the last 22 years on a private farm. The rancher out there provides some passive protection of these populations, and there are literally thousands of prairie rattlesnakes. We have collected as many as 100 at a time in about 25, 30 minutes. One of the ranch hands came out with us. His wife was out there, took some photographs, posted those on Facebook, and she got comments back, oh, you should pour gasoline on that and burn all those damn things out, which is kind of a common attitude towards venomous snakes in particular. Well, she had been talking to Kay, my wife, and she had told her a little bit about some of the things we're trying to do in the lab with some of these compounds. And the ranch hand's wife just railed on this person. Don't you realize there are compounds in their venoms that might in fact be useful as therapeutic drugs? And if you kill these animals out, you're killing off a possible cure for a disease that could be affecting you. And so it was a really potent demonstration to me of just what you can do with outreach and communication to people if you can bring it into the realm of their personal experience. And I could go out there and talk to them about all the nuances of biochemistries of these particular enzymatic components and all. Hey, it looks cool. Look at this three-dimensional structure of a molecule. And after they woke up, they might ask me about something else. But if we can use those kind of hookers to get into it, and that's what it took for me to gain an appreciation of chemistry, for example, something that was now in the realm of my own interest and experience. So kind of going from that point and maybe even trying to answer your question.
It's nice of you, Steve.
Yeah, well, I try to be considerate on occasion, just step out of character, So what do we need out there? We think we know a lot about things like natural history, and I've heard some comments, one of which was to my major advisor during his tenure review, that are less than complimentary concerning natural history type studies and even evolutionary studies that depended on natural history information. But we really don't know that much. We know a lot about a few things, but we know very little about most things. And as an example, we did herpetological surveys in the 90s of Colorado. Colorado has very large counties. And in the space of about 3 years, we came up with 35 county distributional records. And this is in spite of the fact that people had been working for 100 plus years all across that part of the state and hadn't seen or documented this species or that species in that county. So we really don't know basic information, as some of the people were talking about earlier here, as far as does it even occur here? And so in the context of a changing environment and global climate change and human impacts and all that, having that baseline information is critically important because we don't know what we lose if we don't know what we have. And so, so I really think there's still a very strong need for that kind of basic information. And if we look globally, it becomes even more profound because we know even less about most species that don't occur in the United States and Canada and Western Europe and parts of Mexico and places that we've been kicking around for a long period of time. So,
I'm going to switch subjects, stay in the same general area. But first thing I'd say is on your website, there's a great podcast episode, and I believe it's through the university, correct?
Yeah, they put it together.
Where he talks about a lot of these things. He talked about scent trailing and some of these other things. But maybe you could just kind of give the audience an overview. You talked about how you can really link people to some of these concepts by bringing them the things that have meaning in their lives. And so you use the example of venomous snake bites and how that can obviously make them sick, impact their life. But one of the other things that I found was really interesting on that podcast I listened to was a lot of the work you're doing with snake venoms and how that has potential for humans, say in the human health field, for example. And I was wondering if you could just kind of give the audience maybe a little bit of an overview of the work you've been doing in that space.
Well, we've worked with a couple of different compounds. So one, I had a visiting scientist for a year working in my lab from India. And in India, Daboya is a big problem. It's the Russell's viper, and that causes horrific outcomes for people. And if it doesn't kill you, the morbidity associated with a bite is terrible, tissue damage that's permanent and things like that. So he was very interested in looking at that particular snake's venom, and it turns out it's very complex as well, has all sorts of cool molecules and enzymes. But we worked with one of those that interferes with blood coagulation. And it turns out that a lot of the rattlesnakes have compounds in their venom that inhibit blood coagulation. And you think, well, why would that ever be useful as a therapeutic? Well, there are certain scenarios, such as major surgeries, where you don't want blood to be clotting. And so some of these compounds, and they're called thrombin-like enzymes, some of these compounds go in and they take one of the first molecules that's involved in making a molecular patch of your blood vessel when you get cut, and they hydrolyze that compound. They cut it in particular places such that it doesn't form that nice patch. Instead, it forms what are called microclots, and they're basically tiny, tiny clots that circulate. The end effect is that in a very serious envenomation, especially from something like a Russell's viper, you get blood that won't clot at all. And so even trying to put an IV line in a person, you can lead to serious bleeding just from that simple puncture like that. So there are compounds like that that may in fact be able to be purified from the venoms, probably transferred into bacteria, cloned and overexpressed in that sense. And then we can produce them as a biological drug of sorts. And there's been some use of those also in some other applications as well. One of my favorites along that line actually comes from the Gila monster. And the Gila monster has an enigmatic venom as well. Most of the venoms are designed because the snakes are feeding, or the reptiles are feeding on larger animals, and so if a rattlesnake envenomates, it can incapacitate and drop prey very rapidly. No physical interaction. Well, Gila monsters feed on eggs and nestling animals, and neither of those put up much of a defense. So they're probably using it for a defensive reason. Coming back to our application, a number of years ago, a private lab purified and characterized a little peptide compound from Gila monster venom that for certain type 2 diabetics became essentially a miracle drug. And it quelled appetite, and it also helped to regulate blood glucose levels, blood sugar levels. And so that compound, for that company, they made about $250 million when they sold it to a major multinational. So that generated a lot of interest in looking at venoms as a source of biotherapeutics, that there are compounds in these venoms that if we can characterize them properly, purifying them from all the other components, they don't cause problems, but we can actually use them therapeutically. And a lot of the venomous animals are being looked at for those kind of compounds right now.
Great, thank you. So I think we'll take a few minutes and open it up. Does anybody in the audience have any questions for any of our panelists? And if you do, again, raise your hand because we're going to bring a mic over to you so it can get recorded. Josh? Or you had a question, Mark? If you have the mic if you want to go first.
Okay, I'll go ahead and go first. So this is a question for Lisa. And Lisa, as you well know, one of the very basic aspects of snake natural history is their shedding. And one of the things that you commented on that I think is potentially pivotal is the non-random aspect of shedding that you found in whip snakes. Could you comment on basically what that is first, and also how it translates to discovering other aspects of its life history, and how it also translates to discovering aspects of some parallel things in other snakes?
All right, so I do a lot of my work looking for shed skins, and I'll just go back. My professor, when I was in grad school, used to walk in from the field and throw shed skins on my desk and say, "What is this?" And so that's how I started, and would figure that out. And so I've always had an interest in the shed skins. I look at every single one I find. I'm always curious about where they are and when you find them. So snakes shed at different times. Sometimes it's individuals. A snake can shed many times during a season as it grows, but there's also times where there's some synchronous shedding. So after the females lay their eggs in some species, or around the time they lay their eggs, before some snakes enter the hibernaculum, they shed. So you can learn a lot. And it also is a way with so again, with the striped whip snakes, I had such difficulty finding them because they're so rare in Washington. And what was taking us about 8 hours to find an individual snake, we could find a shed skin in about 2 hours. And so we switched to that method and started covering large areas, looking for the shed skins, and then from that started figuring out more about what the snakes were doing. And so what Mark was mentioning is that there was quite a bit of fidelity in terms of where those sheds were found. And you could go back to those, not necessarily every year, but the same exact locations used. It can give you a sense of what is important habitat during what seasons, as well as just where the snakes are on the ground and how common they are. It's a little bit hard to know because we don't always know if all the shed skins are on the surface, or if sometimes the snakes are shedding below the surface, like what proportion of the population is shedding. But for a very rare species, it starts to give you some information without touching the animals or doing anything to them about what's happening with the population. So, did that.
Go ahead, Josh.
Right, I wanted to call attention to some of that proactive sterilization stuff that we were talking about earlier. When Steve and I had to apply for lots of permits in the Mountain West states, really most of the western states, seemed like most of them, if not all of them, required a very clear protocol for sterilizing equipment and such between sites.
I'll just echo that. I've seen that. I get a wildlife permit for amphibians, and it was 2 years ago, I think, that I first saw some disinfection mandates there, and hallelujah. The protocol that my permit is referencing was developed at Southeast Partners in Amphibian Reptile Conservation, Southeast Park, and they have just updated that protocol. And so a new one is being put online. I'm not quite sure if it's online yet, but the big difference is that they've upped the concentration of bleach. It turns out that B-sal is not easily killed by a 10% bleach solution, so you have to increase it a bit. And so I think we'll get that news out shortly once the website is updated and posted, and we'll try to get it out in a few other places as well. But I want to just shout out to those folks. I think it was Jim Julian and Molly Bletz and others at Southeast Park that have developed that disinfection protocol. That was Southeast USA, and now it's being used across the United States and mandated by the state agencies for scientific work. And it's relevant for snakes, even though it was developed for amphibians. So, yeah, great. Thank you for mentioning that.
Great. Anybody else have any questions for any of our panelists? Okay.
I just wanted to follow up on that comment of disinfection, it's the kind of thing that if you leave it out of your protocol proposal, it comes back to you for refinement. So it's a good thing that this is being brought up. Plus, I mean, there are other diseases that are turning up as well. So hemorrhagic viruses and logomorphs in particular, jackrabbits. We're also made aware of that through the scientific permitting process as well.
Yeah, I did sort of an advertisement in my talk earlier today, and I mentioned the pentostomiasis, the pentostomes. It's a snake lung parasite. And I didn't read everything that was on that slide, but pentostomes infect snakes and mammals, and we do not know if it's zoonotic, meaning we do not know if it could be infecting humans as well. So that work has not yet been done. But
Finding lots of them in indigo snakes, by the way.
yeah. Oh, I'm sorry to hear that.
Including our captive. We have a captive breeding facility for reintroductions, including
So yet another reason to be careful with the disinfection, but if you're handling animal to animal that have pentostomes, then there's a reason why you would be wearing gloves and changing those gloves between animals as well.
Mm-hmm. Okay, great. Question mark? Go ahead.
Yeah, this is a question for Lameace. You mentioned a diversity or a range of outreach conditions of different kinds. In your perspective, what do you think is the most successful approach to outreach in terms of what you've seen, although I know this kind of thing probably hasn't been evaluated very much yet?
Bringing in live animals. Like 100%, like having live animals, like whether it's at like farmers markets and stuff, like really, we have found a lot of success in not just reaching kids, but also reaching their parents or reaching, you know, like landowners who come by, who they tell us whatever story they want to tell us about killing the same snake they saw on their property, but it's still like it sparks a conversation. And so I think our best outreach programs that we have are just like hands-on programs where they can actually like see the animal.
Thank you.
Mm-hmm.
Okay, so I'm a collector of snake stories. Lameace well knows this fact. So Lisa has generously taken us out on an excursion looking for striped whip snake sheds. And we're sitting around the campfire at night in the dark. Maybe we have a few cold beverages going. And we'll go backwards now. I turn to Steve and I say, tell me your best snake story. Just like imagine you're with your fishing buddies and you're telling them about that big fish you caught.
You mean the one that I made up, or that I
Take it away.
I'm not sure that I have any really unusual and unique ones. They're probably things that you've all heard reiterated. And one of the common ones that we hear are, that rattlesnakes are losing their rattle, so they can't warn us anymore. They're going to sneak up on us now. And part of that is because they're hybridizing with bull snakes. And I actually had a person that was head of the animal facility at a major university ask me if that was true. And this is a well-trained veterinarian. So those are quite prevalent. One of my favorites.
I want to hear your snake story.
Oh,
no, I don't want to hear like some mythological, and we all know those. I want to hear like the time you were 10 years old and grabbed a rattlesnake or whatever it might be. What's your best snake story?
Well, actually, I wasn't 10. I was 15 when I was bitten by a Southern Pacific rattlesnake, but that was incidental. It only put me in the hospital for 6 days. But the more interesting one was I was working as a high school student for a company in Southern California that imported and exported reptiles and amphibians from all over the world. We saw everything you could possibly imagine. I was in charge of the venomous snake room, which was about 10 by 10 feet and had cages to the ceiling and on all 4 walls except for the door. So there were hundreds and hundreds of snakes in there. So one of the charges were 3 king cobras, 2 of which were only little guys. They were only about 11 feet long or so. And then there was a big one that was about 14 feet long. And so I took them out of the cage and put them in just a regular plastic trash can, 55-gallon type trash can. That's our holding tank where we're cleaning them. Snakes number 1 and 2, I got into the container without too much trouble, but snake number 3 would have nothing of it. And so for the next 10 minutes or so, we went through this duet where it was hooded up about 4 feet off the ground and it would charge at me with its mouth open, and I would retreat and close the door. I did that several times, and the last time I didn't quite get the door closed fast enough. It was a Keystone Cop adventure because there was another idiot that worked there that was watching the whole thing. We had a double door scenario, and so we're kind of tumbling over each other, trying to get out of the way of the snake as it came into the main room. So we got it into a smaller room off to the side where the temperature was colder because it was for amphibians. And that's when I decided 2 things. One, probably this is above my pay grade of $3 an hour. And 2, it's time for the boss to take care of this issue. So he ended up catching that, pinning it down, and getting it put away. So that was kind of a personal experience, one that was, caught my attention.
King cobras always make for good snake stories.
Oh, yeah.
Lameace, tell us your second best snake story. You can tell us your best snake story again if you'd like, but tell us a snake story.
Yeah, so I've told a snake story before on Chris's podcast, but I'll tell it again because it's I think just like one of my favorite stories, and it involves Lisa actually. So I was helping Lisa do some of the striped whip snake shed sites, and she rolled out this big map, and she was pointing out all the sites to me. And there was one in particular that I wanted to go to, and she kept warning me that there were a tremendous amount of rattlesnakes at this site. And I was excited about that. Like, that didn't deter me. And so I'd gone out there quite a few times. I hadn't seen a magnificent number of rattlesnakes, but there were quite a few. And I ended up finding where the entrance to a habernacular was, and it was close to a striped whip shed site. And so before I moved down to Florida to start my PhD, I went back to the site one more time, and it was super early in the morning. And I remember going around to the entrance of the den and jumping up on a rock, and the whole side of the rock just erupted in rattles. And it was the most deafening thing I'd ever heard. But I couldn't see any of them. And so it was a little bit of a moment of just kind of terror of not knowing where to look. But as I kind of focused in, there were rattlesnakes everywhere. And after counting them, there were about 150 around the entrance to the site. And big females with little neonates on them and stuff, and to me it was just a good send-off from Washington. But I remember going back to Lisa and being yeah, there are a lot of rattlesnakes at that site, and she was yeah, I told you. But yeah, there was no whip snake in that whole group, but there were a lot of rattlesnakes. So
That sounds like a good time.
yeah, it was fun.
Dede, tell us your best snake story.
So it has a 2-part. So the first part is I don't remember the first snake I ever saw because I think I grew up as a free-range child, and I encountered these things early before I had memories, really. But there's a photograph of me. I think I was 3, and my brother, who's 2 years older, so he must have been 5, was there with 4 of his friends, and they're holding a snake behind me, and they're all just grinning and everything, and I am crying my eyes out in front of them. And I don't remember the incident, but we were out catching stuff all the time. So now flash forward, and I wanted to do things that Lameace is talking about to get my kids used to animals and not be afraid. Mm-hmm. And a garter snake appeared in the yard one day, and so opportunity. This is the first snake I think my kids have seen in nature, or the yard, I guess that's nature. And the neighbor girls were there, so there were the reverse situation of several girls, and then my son, who must have been 3. and they're all standing around the garter snake and like, oh, what are we gonna do? And I felt success when my son reached out and touched the tip of the tail of the garter snake, and it's like, yeah, he's not afraid. And then he picked it up and whipped it around his head. Woo! And all the girls started crying. And I'm like, that might have been what happened to me.
That's great, Dede. You ready?
I'm ready.
You're going to take us home.
All
Do
right.
it, Lisa.
So I'm not going to tell a striped whip snake story. I'll tell a rattlesnake story because those are the most exciting when you're in the field. And so I was radio tracking rattlesnakes. And for those listening, when you're doing that, as you get closer and closer, the sound gets louder and louder that you're hearing from the transmitter. And so with rattlesnakes, it's kind of exciting because you know there's a rattlesnake. And it's not exact, right? You're getting closer, and sometimes the sound is coming and going. So I had narrowed this down to a shrub, and I'd been tracking the rattlesnakes for a while. And so I was expecting the snake to be on the ground somewhere underneath the shrub. And so I'm searching and searching, and I'm getting closer and closer. And sometimes as you get really close, you can't use your antenna. So you take off your antenna, and then you're doing weird things like dangling the cord and moving things around because you're trying to triangulate to where the snake is. And the whole time I'm looking at the ground, I'm trying to figure it out, and can't see what I'm doing. And eventually I'm moving up higher and higher and dangling this cord. And I look up, and right next to my hand, up around the level of my eyes, the rattlesnake is up in the top of the shrub, sitting there. And I have been fiddling around right next to it, and the rattlesnake didn't strike or anything, nor did it rattle. And when you're doing telemetry and you're following these, so that scared me, by the way. I had the moment where I'm like, but it is amazing when you're tracking them. They don't always rattle, they don't always strike. A lot of times you're working around them and they're just kind of doing their thing. So that for me was one of the most exciting things because it was so unexpected. I really had thought that the snake was on the ground and not up at the top of the shrub where my hand was.
So we've got 3 venomous snake stories and a garter snake rodeo. So first of all, let me thank all of you guys for being here today. And I wanted to, Mark, you had one final thought?
I just want to put a little exclamation point on your snake stories, because I've got a good one for you if you want it.
I do, got 2 minutes.
Okay. So when I was in Costa Rica, I was measuring and weighing every snake I could possibly get my hands on. And one day I got a juvenile palm viper, and I was measuring it on the table and being very delicate with it, but it was very squirmy, and it squirmed out of my left hand, and it struck at me, and I pulled away as fast as I could, but it hit me on the tip of the index finger. And of course I was shocked, and I looked at it, and it had pulled out one of its fangs, and as I looked at it, it hit a blood vessel, and I could see the blood coming back up through the fang, and basically, of course, I pulled it out right away, and I realized, of course, that it had been a dry bite. And it gave me a perspective on dry bites in pit vipers that, for many of the species, as long as you don't really harass them, they're striking basically defensively and not injecting venom. I'm not suggesting that that should be something that you take for granted and go picking up pit vipers, but it did change my perspective on it.
Great. Well, we're going to wrap it up there then. Again, thank you guys so much. And I just wanted to thank all of you for being here and everybody else out in podcast land that's listening right now. Thank you so much. And I just want to tell everybody to remember, snakes are animals too, and it's a privilege